So we were going to do a panel discussion about the research that we're doing in genomics all the way through. But then we realised that you guys have absolutely no context for what it is that we're doing or, understanding it. So what we're actually going to do is a couple of short presentations to get you up to speed on genomics and stem cells, and then we'll do a panel discussion. So hopefully this isn't too much for after lunch, but by the end of this, you should understand a whole lot about genetics. That's my hope anyway. Don't stress too much about what's on the slides. They're for people who are visual learners. There'll be a few in the audience who can only take it in that way. Mostly you can ignore the kind of stuff on there. All right, so this is going to be a bit Coles to Newcastle to start with. We know MS is a complex disease, it's a neurological disease, and it results in progressive disability. Some of the things that are important for the genetics part of it are it's an autoimmune disease, so the body incorrectly attacks some part of itself, but it also has this underlying nerve damage or neurodegenerative component. We know immune cells enter the central nervous system and they attack the myelin. And if we want to have a look at a picture of that, you can see the nerve cells are the long things with the little frilly ends, and the blue bits are the myelin, and the blue cell is the oligodendrocytes that are putting that myelin, like wrapping those cells, keeping them happy, and pulling that myelin in place. So that's the bit that gets damaged, and that's the bit that we're most interested in fixing. Not so worried about fixing the immune side of it, we actually want to fix this because that's where the real problems come in. So now we're going to get into the genetics part. So we talk a lot about the human genome. I'm going to explain what we mean about that. So this is your underlying code that creates your entire body, right? This is the information that your body uses to make you. And everybody's is going to be different, but there's a lot of similarity here. So the code is made of 4 letters, A, T, C, and G, that's it. One human genome is about 3.2 billion of those letters in a string. They're in a particular order, and they're spread across 23 chromosomes. So they get broken up into sections, that code. Only about 3% of all that information actually codes for genes or the bits that the body uses to turn into proteins and to make things. So the tiny amount of that whole overall genome is what we call coding. That's where the kind of core information comes from. If we were to think about the human genome as a physical thing, so think about it as books, the Wellcome collection actually printed a copy. So it ends up being 120 books with 1,000 pages each, and that's at 4.5 point text. So I can't read that. I can't imagine anyone else could. But that's just one genome. So we've got 2 copies of that. All of us have got 2 copies of that. This is what we're looking through when we're doing genetics research, right? So they talk about a needle in a haystack. We're looking for letters in amongst all of these books. So why is genetics important in all of this? Because your genes control everything, your basic biology, how your body works, how it makes teeth, how it knows to put teeth where teeth go and not putting them coming out of your head. It also controls how you respond to the environment or how you respond to individual drugs. We talk about personalised medicine. That's the things that are unique about you that change how you might respond to treatments. So your genetic changes, everybody has genetic differences between each other. Your individual changes make you who you are. If we didn't have them, we'd all just be a group of male and female clones. But when you think about that, each one of us is only about 0.1% different from each other in our coding. So we're incredibly similar. And then we have this tiny little difference that makes us all unique, right? These kind of changes, while making you, can also have other effects. So some of them can be the cause of disease. Some of them can influence how you respond or don't respond to a particular treatment. So they're pretty important. And what we do is we study those differences between people, targeting particular things. So we want to understand how to improve health or how to improve treatments, or whether there are different treatments that we haven't picked up before based on genetic changes. Okay, so that's basic genetics. I want to talk about genome sequencing. And that's when we in the lab want to read your genetic code. We want to look at your string of letters and see exactly what's there or what's not there or how you're different to somebody else, right? So we've got technology that now lets us read that entire sequence. And we do that by looking at a human reference genome. So we've got this mishmash of all these different people that we stuck together that gives us a set of those books that we use to compare all the other books to. That's our reference individual. It's not one person, but it's like the most common genetic variation at each of those points. So we compare a sequence from an individual who we sequence, so it might be one of you guys, against that reference genome, and then we just look for all the places where there's differences between those two. So we're not interested in all the places where they're the same, we just want to know which bits are different. So remembering that's about 0.1% for any individual, that's about 3 million places in those sets of books where there's differences. Not many, no, not many, it's tiny. We've got ways to make that smaller. So, like I said, we only focus on where it's different. So that's like taking all those books, we rip them up into shreds that are quite short, and then we line them back up against the original set of books, each of those short sections, to try and work out where they overlap. And most of that shredding will overlap perfectly, and then there'll be a couple of places where there's one letter difference. And that's the ones that we want to focus on. And we do this multiple, we shred multiple books so that we get a number of strands each time that overlap to make sure that the changes are real changes and we haven't just introduced some kind of error. Okay, so let's go back to MS and genetics. We know that there is a significant genetic risk in MS. It's not complete. It's not like one of those single gene disorders where you get one mutation, you're going to have the disease. It's much more complicated than that. And we also know there's environmental factors as well. So genetics is part of it. We have some inkling of how those things work together, but that's way too complicated. So we're just going to focus on the genetic component for now. So I'm going to do this analogy, which isn't 100% accurate, but hopefully you get the idea. This is how you think about a complex genetic disease. So this isn't one where you've got one genetic mutation, you get the disease, right? In MS, there's a lot of different genetic variation. And say you have to get up to $100 before you have the disease. You might have that as a $50 note and a bunch of small change, or you might just have an entire wallet full of pennies, right? So each of those ones is a genetic variant and they have different levels of impact. So obviously a $50 note genetic change is a really big one. That's going to account for a lot of it. Your 1 cent pieces, not so much. You're going to need bucket loads of them before you get to your $100. So no one needs to interpret this picture except that it's incredibly complicated. This is what we know about the genetics of MS so far. So around the circle is the whole length of the human genome, and then each of those spiky bits is a place where we've found that people living with MS are more likely to have a genetic change at that site than people without. Okay? But in terms of the overall picture of MS, so there's 200 of these, 200 changes across the whole genome. The overall picture of this whole circle is what we know is the whole genetic risk for MS, and that chart, those 200 places in the genome, account for that small wedge of it. So we get a lot of press about, we've identified all this stuff in genetics of MS. It's a really small part of the overall. So our research at Menzies takes a different approach to this. We're not as interested in those changes in the genome that are common and people share, because they, in terms of that wallet analogy, they're the 1 cent pieces. They're not giving you any of those big indications of what's going on in genetic change. So we're interested in families with MS. So families where you have multiple people diagnosed are fairly uncommon, but they do exist. The reason they're uncommon is partly due to age of onset. People find out early on, sometimes they don't go on to have children, but we do get these multi-case families, right? Closely related individuals, so I mean parents, offspring, siblings, tend to share a lot of what we call the common risk exposures for multiple sclerosis. So they tend to all get glandular fever in a cohort, tend to have similar sun exposure profiles based on where they live and the kind of activities they do as a family. And they also share the common risk variance, because in families, so all of us are 0.1% different to each other, but families closely related, parent, offspring or siblings, share half of their genome with each other. They're way more common in terms of the kind of variation they have to each other than they are to anyone else in the general population. So in terms of those common risk variants that we talked about, families will tend to share most of them. Whether or not the individuals have MS or not, they all have the same common genetic risk profiles. So we're interested in looking at these families because we think what will tend to happen is that the rarer genetic mutations, the ones that you don't see shared across the general population, do tend to segregate in families. They do tend to get passed on in families. So if a founder of a family has a rare mutation appear, they will tend to pass that through the family to multiple people. So by looking at families, we can try and identify some of those genetic changes that are not seen in the general population, and we'd never find them if we studied the general population. But if we can see them in multiple people in a family, perhaps it's all the relatives in the family who go on to develop MS have that mutation and their unaffected siblings don't have it. So that's the kind of premise of what we're looking at. And because those variants are rare, not seen in the general population, they're the kind of things that you expect will have a big impact on genetics. So they'll be in those coding regions, they'll be influencing how the gene actually behaves, having some major kind of biological impact. So this is quite detailed, but to give you a basic overview of the whole pipeline, and this is what we'll talk about as a panel discussion, we recruit people to the study, people who have these kind of families where you see in black is the people who've been diagnosed with MS, white's the people who are over 50 and haven't developed MS. We recruit these kind of families, we make stem cells from them, we look at their genetics, and then we do some work in the stem cells, which Kayleen's going to talk about. We eventually will hope to get to the point where we can do some drug screening. So if we find something that's got a known drug target, we can immediately go and look at those things. And then we've got this pre-established pathway for clinical trials. So this is with Bruce. We've already got processes for this. So if we identified something, we've got a really speedy way to get it through to clinical trial. And then the cycle just keeps going. Okay, so these are the kind of families that we're looking for. Where you have 3 or more people, closely related people, so parents and their children. Most of you would have seen family tree diagrams before if you do family history and things like that. This is the kind of thing we're looking for. So affected multiple people who've been diagnosed with MS in a family. It's a terrible thing, but it is something that can massively advance our research as well. So in terms of the analysis, we've got people like Nick, who you probably would have met out on the trade desk, who looks at this genome sequencing and he compares the individuals who've been diagnosed with MS against their siblings. So not only is he looking for places in those book collection where you see the genetic changes, he's then narrowing it down to, okay, what is it that these individuals share that their unaffected relatives don't have? And that can really quickly lead us from looking at 3 million places in the genome down to maybe looking at 10. It's a huge, huge shift. Kaylin will talk more about this, but we can then take that genetic information we identify and look at what's the actual biological impact of that. And we can do that in cells that we make from the family members. So she'll cover all of this stuff. But it's huge. It's such a massive change in the way we've done things before. We've always used big population studies, and then we've gone to mouse models, and then, it takes 25 years to go from discovery to a potential for a drug target, let alone an actual drug target. This can massively shorten that time frame. I might skip this bit because you're going to cover, wherever you've gone, you're going to cover all of the MS STEM, right? So I'll let her do this stuff. But basically, we take a stem cell, we can make oligodendrocytes from those stem cells, and then we can study those actual human oligodendrocytes from individuals who've been diagnosed with MS in the lab. So yeah, these are the cells, but she'll cover that stuff. So in terms of recruitment for this study, when we identify a family who is willing to participate, all we need to do is take blood. And see, it's just a blood sample for the genome sequencing and a blood sample for making the stem cells. We get to turn those stem cells into cells that are relevant to MS, like oligodendrocytes. We look at the way they behave. So if we looked at individuals with and without MS in this family, we could look at the differences between how their oligodendrocytes behave. Then we can go to that genetic mutation and we can either put it into the cells who don't have it or take it out of the cells who do have it and then look again at how they behave. So it's a really cool bit of science. So overall, this research is basically using a personalised medicine approach. We're looking at the individual level differences about people that might regulate their disease or change their disease outcomes. So we use the families to pull out that genetic signal in the first place, and then we can go back to those big case control population cohorts to say, okay, what does the general MS population look like at these points? Then we can study the actual biological role of the genetic changes that we identify in cells that are actually relevant to MS from the people who they're relevant to. So not mouse models. So it gets us to that really precise biology of what's going on in MS at a personal level, at a per individual level. There's flyers out in the trade hall for recruitment for this study. If you know of any families who you think might be appropriate and might be willing to participate, please grab one and pass it on to them. That would be fantastic. It's definitely the hardest part of this study is identifying these families in the first place. And genomics is incredibly expensive, so we've been hugely fortunate to be so well supported, both in the collaborators for this project, but also in the funding side. Thank you.
Awesome.